The overall goal of this procedure is to perform a craniotomy on the ventral skull of rat pups for in vivo electrophysiology or two-photon imaging studies. This is accomplished by first anesthetizing the rat pup on a heated pad and using a ventilator to control its breathing. The surgery begins with exposing the ventral side of the skull, followed by a craniotomy to expose the brainstem surface. This procedure sets up the use of in vivo electrophysiology and two-photon microscopy to analyze structural and functional aspects of neural structures.
The main advantage of this technique over other methods, such as acute or organotypic brainstem slices, is that it provides a system to study the dynamics of diverse cellular processes in the mammalian brainstem in vivo.
Individuals new to this method will struggle because the rodent pups are small and the methods assume some proficiency with surgical technique.
This surgery requires mammalian Ringer's solution, the proper tools, a heating pad, and a small animal ventilator. Take a confirmed anesthetized animal, position it dorsally over a heat pad set to 37 degrees Celsius, and attach the nose cone to sustain the anesthesia. Tape down the limbs and tail, and then clean the skin using saline or disinfectant solution. Now make a longitudinal cut through the neck epidermis. Then make 4 lateral incisions.